Hi, welcome back. This is the second in a short series of videos around using a centrifuge. As a quick recap, in the first video we went through the principle and components of a centrifuge, and today we're going to talk in a bit more detail about how to use one and how to balance a centrifuge. So let's get started. So firstly, we're going to use the centrifuge to send blood samples which are collected in the community by venipuncture, and the people who are going to spin the samples are yourselves who are collecting the bloods. And we briefly discussed why in the last video. It helps reduce the number of rebleeds and improves the accuracy of the results we can produce. So first of all, what samples do we put in the centrifuge? To answer that, when you're asked to collect the blood samples, we need different types of samples for all the different tests that we do. So you're probably asked if you're using the BD vacutainer system to collect yellow tops, purple tops, rust coloured tubes, blue coloured, red. There may be some others, I can't quite think of them. But the ones that you are going to place in the centrifuge, and the only ones you place in the centrifuge, are the yellow top and the rust top. These are serum samples, so we need to separate the red cells and the clots from the actual liquid part of the blood, which is the serum. Both the rust top and the yellow top in the BD vacutainer series have separator gel in the bottom of the tubes, which helps with the centrifugation process and helps remove them. So to show you the tubes that you are going to centrifuge, I've prepared some, so they're over here. So let's have a quick look at them. The BD yellow top serum tube. This one has a sample that has been collected, so it has blood in it, and the clot has been allowed to develop over 30 minutes. The bottom of the tube has the separator gel. So that is a tube we are going to ask you to centrifuge. The second type of tube we are going to ask you to centrifuge is very similar, except it has a rust-coloured top. And the rust-coloured top goes the whole way through the lid. And again, it has the separator gel at the bottom, and this has a blood clot in it. I'm going to point out that the rust colour can look a bit like the red colour. The way you can tell is that the red is actually a bit redder, has a black middle, and the red top does not have any gel in the bottom of the tube. So quick recap, the only blood samples that you should place in the centrifuge and spin are the yellow and the rust. Okay, so how do I centrifuge these samples? Well, in the first video we discussed the components, so we should be familiar that we have brackets. Your brackets will contain 4 or 5, if I have the right ones, 5 slots for samples. We take these out of the centrifuge buckets, and you can load your yellow tops and your rust tops into the brackets. Now, we must talk about balancing. Centrifuges generate centrifugal force and they'll spin very quickly. To work properly, or to work at all, they need to be balanced. So there needs to be an equal amount of weight, approximately an equal amount of weight in each of the buckets, or definitely facing each other. We call that symmetry, or the mirror image. So I'm going to show you how to balance a centrifuge now, and this will stop it vibrating in use and reduce the amount of noise and reduce the wear and tear on the centrifuge. The centrifuge actually has a sensor in, and if it isn't imbalanced, it will stop and beep at you, and you'll have to open the lid and balance it and restart it. So let's just go through that. In this example, I'm going to centrifuge one sample, but we can obviously fill the centrifuge up to the capacity of the amount of brackets, sample holders that you have. So your centrifuge can hold up to 25 serum samples, and you can spin as few or many as you like, but it must always be balanced. So I've placed my one yellow top into a bracket. I place that into the bucket. I place my empty brackets into the buckets to help distribute the weight and make sure the weight is spread. We will also supply you balance tubes. So these are here purely just to create the symmetry that you need and to distribute the weight of the centrifuge. We will supply you balance tubes that are a different colour to the ones you're going to send to us, so that there can be no confusion that you place your serum samples in your balance bucket and your balanced tubes don't get sent to the laboratory. I place one sample or balanced tube opposite my sample. That has symmetry, so in effect the centrifuge is balanced. For good practice, we also suggest that there's further symmetry so that there is equal weight distributed against the centrifuge. If I wanted to spin two samples, I would place my second sample in the centrifuge, and to get balance, I would place a balanced tube opposite. I'll take one out. Once we're happy the centrifuge is balanced, we can start the spin cycle. As we discussed last week, this will be for 10 minutes. So let's go through that again. We've balanced the centrifuge. We check our buckets can rotate on their bracket. We place biolids firmly on the centrifuge. We check the rotor for debris. We close the lid. And we press start. And the centrifuge will display the countdown and the RPM going up. Centrifuge will run for 10 minutes. It will vibrate slightly, make a slight noise, but that's fine. If it was unbalanced, there would be more vibration and the centrifuge would shut down and it would beep and you would have to open the lid and check what was going on. At the end of the cycle, the centrifuge will deaccelerate and I will show you what the samples will look like after centrifugation. So this is yellow top, which has undergone centrifugation, and you can see now the blood clot is at the bottom of the tube. The gel separator is sandwiched between that blood clot and the actual serum, the straw-coloured liquid. I can invert the tube and the red cells no longer will mix because the separated gel maintains the stability of the sample, and that is now ready to go to the laboratory. Place that in the bag with all your other samples and send it to us and we'll do the testing. Right, thanks for watching that today. Please recap and watch this a number of times so that you're comfortable using a centrifuge. You can always use the balanced tubes that we've provided to do some trial runs to make sure you're comfortable with that. And then there's a series, the rest of the videos in this series, we're going to cover a bit more detail to allow you how to package the samples and send them to the laboratory, how to deal with a spill, which is pretty rare if you use the BioSeal lids and use the centrifuge properly, some general maintenance tips, some troubleshooting, and then some Q's and A's and how to contact us. Hopefully this has been useful. Thank you very much.